So we'll be talking about SNP calling and what you do downstream of SNP calling. Once you obtain your raw SNPs, how do you filter them for high quality SNPs? But before we do that, can we revise what we talked about on Monday? Okay. Do we have any questions? Specific topics you want us to go back to talk about? So someone mentioned that maybe some people might have issues with like understanding Illumina sequencing. Can we say we're all okay with the way the Illumina platform works? In terms of when you get your reads, you know how those reads were derived, right? In terms of if you're doing a ped-end sequencing, how that physically happens on the machine, and if you're doing a single-end sequencing, how that happens. Okay. So have you seen the video on how it works? Okay, let's watch that video. And there are no stupid questions. So let's go to YouTube. So if you have a question and you think it's stupid, ask it. It might just be a single point that might take you over that hump and get you into a deeper understanding of the topic. So if you have any question, don't hesitate. So
I'll go ahead and start passing around to everybody. I've got sort of a walkthrough for what we're going to do today. And at the beginning of the walkthrough, I've written up a few approaches to pulling up some of the images that we created last class. I know we were kind of just running lines of code. It's all on a command line interface. And didn't get to see the output really. You just saw the output that I had up there. So hopefully the first few lines on here will kind of help you figure out a way to get that onto your own personal computer. If you're on Windows, I've only got a little walkthrough on what to do if you're using PuTTY. I'm not sure what other ways people are connecting through SSH on Windows, but I'm sure there's a way that we can get those files. So let me know if you guys have any problems.
Anyone knows how to turn on the sound on this system? Okay, I think we might be handheld. Okay. Hello? Hello? Nope. All right. Hello. Okay, I'll just play it out of my laptop if that works.
Sequencing and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. If
you get to a point in the video and you're lost, just raise your hand up, we'll stop it and we'll talk about it.
Reduce cycle amplification, additional motifs are introduced. such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of two types of oligos. Hybridization is enabled by the first of the two.
So it's got two oligos, right? Remember your library has two ends, right? Two adapters. One hand binds to the other and the other binds to the other oligo, right? To the blue and purple, right? Or something.
Two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule.
So who knows what those two strands are? The sense strand and the antisense strand. So if you're trying to sequence from the left side, you go with the blue one. If you're trying to sequence from the right side, you go with the purple one, right? But you'll do one at a time, right? So
That are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called.
So what's happening here, you have millions of clusters, right? And each cluster has several oligos, right, sticking out in it. So it's like, just think of it as several pins clustered together in one cluster, right? So you have several clusters. So when you generate that signal, so let's assume you have 1,000 pins in each cluster. Those 1,000 pins will light up together at the same time, right? And if it's a G, they should all be lighting up with a C, right? So because it has several probes in the cluster, that intensifies the signal, right? And then you can use a camera to image that, the color. Huh? So the first few cycles you do, the machine does something that's called clustering, right? So if you think about it, you have millions of clusters. You don't use all those clusters. If you overload your slide with too many fragments, right? Then you have like all the clusters have like all that signal and they're too close to each other. So you want to find that balance where you're not really using up all that cluster so that each dot of light is distinct, right? So the first thing the machine tries to do is do the clustering, which basically means what's a cluster, right? It's trying to map the whole lawn to say, okay, that's a cluster, that's a cluster, that's a cluster, and it will physically tag that location as a cluster. And it will give it an ID so that when you get your reads come with a unique ID for that specific cluster, right? So that's, you do the clustering at the initial stage, right?
The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then...
So Read 1 is the left or the forward, and Read 2 is the right or the reverse, whichever terminology you want to use.
Washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification.
So we're good? Or you want to finish the video? Yeah, so if you're doing a paired end, you're reading from the left, Read 1, and you're reading from the right, Read 2, right? If your fragments are long enough, they won't overlap. If your fragments are small, they will overlap. All depends on what you want or how you've designed your library. And you don't have to do a paired end. You can say, you know, all I want is just a single end read. So you just do the forward and the left. And then once you get to, you do that cycle, you're done. You stop the reaction.
So that means the paired end is actually it's a sequencing reaction. The sequential reaction is you first sequence the forward, then
Yes.
sequence the. It's not in parallel.
No, it's not in parallel.
Is kind of forward, reverse together.
No, it's not in parallel. Because if you do it in parallel, it's going to get confused, right? Because it can only read a cluster once. If you read two things in a cluster at the same time, you're going to be producing two colors or two signals, right? And then it's going to confuse the machine. So you have to do the first one first and then sequentially follow through with the second one. We good? All right. So I can just walk through the slide. I'll just flip through it. And if there's something specific on it, tell me to stop and ask a question. It's not a marathon. The questions you're going to get are going to come from the slides. So understand the slides. I'm not going to give you a question outside what we've talked about or what's outside this slide. Do you understand the difference between the Hamming and the Levingston or edit distance?
One of them is just purely about mutations, for the other deals with end is in trying to keep barcodes reading.
Correct. So the Hamming distance only accounts for substitutions, right? So point mutations. But you could also have indels, right? So the Levenstein or the headed distance is a better algorithm because it accounts for substitution and it also accounts for indel errors. All right. And indels can happen how? Maybe during your oligosynthesis, right? Because you're synthesizing your adapters, right? Those could have error, or during the actual sequencing on the Illumina platform, you could have errors there, right? So it's useful to be able to like account for both type of errors when you're demultiplexing, so you're not mixing up your samples. Okay. Talked about phasing. That's supposed to add one base at a time, but sometimes it might add two bases, right? And then that offsets that strand, right? So you're going to be getting erroneous, right? And basically what happens is now you start getting multiple signals from the same cluster, right? And once you start getting multiple signals from the same cluster, it affects your quality score, right? Because the quality score is based on how pure that signal is, right? It wants to read a red. If it's reading a red and it starts seeing a little bit, a smudge of green inside your red, it's gonna penalize that curve, right? So you can see how, if you start off from the beginning with a phasing error, how you can keep amplifying that error continuously as you go through the cycles. So it's essential that you start off clean, right? And the way to start off clean is by making sure you have enough diversity, you have a variable length barcode, right? To give you a clean start and carry you all the way. Talked about optical duplicates because you have PCR bias sometimes. You tend to amplify smaller fragments faster or better than larger fragments. We talked about recalibration, right? Can you tell me why recalibration is not it's not a really good fix? In the absence of a fix, you do recalibration, but can you tell me why recalibration might not actually fix your errors? 2, why do you think recalibration might not really be an efficient way to do error correction?
It's because it's based on making a guess about whether a SNP is real or not, and we don't have that true information.
Okay, so if you do have a reference genome that you compare, a good reference genome, well, recalibration depends on that, right? So if you have error in your reference genome and you actually scored a base correctly, right, with a maximal quality score, what's your recalibration going to do? It's going to say, oh, it's not good, even though your reference genome is a problematic one, right? We also said, if that's a true SNP in your sample, which is what you're looking for in the first place, recalibration is going to penalize you for finding a SNP, right? Does that make sense? One other thing, at least for me, that I have issues with recalibration, so besides the reference genome and that old bias you're throwing into recalibration, think about it. If you're looking for particular sequence features, right? So you're like, okay, if I see a CCC, I know the platform doesn't do a good job calling that. So I'm going to penalize that sequence if it's a CCC. Well, so everywhere in the genome where you sequence a CCC is going to automatically have a lower quality score, not because it's wrong sometimes, but because you feel like you're not confident calling that. Well, what about if the actual sequence was a CAC, right? Is that us? Okay, so that's probably outside. So,
what are the basic conditions for calibration is needed?
so I know this is a bioinformatics class, but the best way to fix a problem is not to have that problem in the first place, right? So if you're making a good library and you're doing, making a good library on the bench and you're looking for wet lab solutions, then you don't need to deal with that problem if you solve that problem upfront, right? So some of the things we're going to be covering in this session is going to be cases where you've used wet lab protocols, right, to resolve the problem, and cases where we're also using computational approaches to resolve some other problems. So think about it as, finding every possible way to resolve a problem and not just using one single approach. Right, so I was saying, if you're penalizing something for being a CCC, what about if the C in the middle is actually a mutation? Right, so I'm just saying that there's a lot of bias that goes into a recalibration correction. And then we talked about how you can actually use information at the beginning of your read, right? So if you know your read has a restriction site, right? If your library was based on restriction enzyme digest, you can use that information if there's a mutation in that region. Remember, we know that you tend to accumulate errors downstream if you start up with error, right? So if you can catch that error that's happening at the beginning and pull it out, then you can efficiently pull out. In fact, in that case, you're not even trying to do error correction. You're basically filtering out the error, right? And we can see how this goes from, this is more obvious, this goes from really somewhat bad to good, and this goes from somewhat bad to really, really bad, right? So that tells you you're efficiently pulling error out and, withholding error to get better calls. Okay. In the absence of any question, we'll move on to SNP calling. So for the SNP calling, we're going to be talking about alignments. We're going to sort of touch on SNP calling in polyploids and diploids. I'm assuming most of you might be working with diploids, but every now and then you're going to deal with polyploids. We're going to talk about the post-alignment quality control and the post-SNP calling quality control and how to remove problematic SNPs, right, from your data set and just keep the really good ones. So we're going to start off with a workflow. And I sort of like the picture, because pictures tell 1,000 words. So the first thing you're doing is the read clipping or trimming, which we talked about. And then we talked about error correction. I would prefer to do error filtering, right, rather than correction. then you do your read mapping, right? So you take all these small reads, right? And you map it to your reference genome, right? And then you remove the duplicates. Did we talk about how to prevent duplicates? How do you prevent or minimize optical duplicates or PCR duplicates? We talked about it on Monday, and I talked about it again today. I know it's a lot of moving parts. Something about PCR bias. I thought I just gave you the whole answer there. How do you prevent or minimize PCR duplicates? Keyword, PCR bias. Okay, how does PCR bias prevent or minimize optical duplicates? Yeah. Huh? Size selection. There you go. Thank you. And what does size selection do? It narrows the size range of your fragments so that they get an equal chance of being amplified, right? If your size range is 1 kb and the lowest is 30 base pair, right? You're going to be getting so much product or amplicons from your 30 base pair, and you're not going to be amplifying your 1 kb that efficiently, right? But if you size select and you put everything between, let's say, 200 and 400, then you've dramatically reduced that effect of PCR bias, because now everything gets about equal chances of being amplified. All right. Then you do realignments, you do your base quality adjustments, which is the recalibration again, right? And then you do your snip calling, and then finish up with filtering. When you do snip calling, people just call it snip calling, but it's not really snip calling. It's a variant calling, right? Because you're also capturing indels, right? You're capturing copy number variation, right? Think of SSRs. Do you know SSRs, right? You have ACC. Sometimes you might get multiple copies of ACC in tandem. ACC, ACC, ACC. So you might have two individuals that have 5 copies of ACC. Another individual has 20 copies of ACC. There you go. You have an SSR polymorphism, right? Sequence repeats, right? So that's a type of copy number variation. You can have the presence or absence, right? We talked about how when you're making your library, sometimes your restriction sites might be polymorphic so that you can't digest it, right? So you won't see that fragment. If you have a good algorithm, you might be able to go in and determine that that's not really a missing data, but that it's actually a presence absence variant, right? And then you have epigenetic markers. If you're using a methylation sensitive enzyme, it's only going to cut regions that are not methylated, but regions that are methylated remain uncut. Again, it's a little bit messy trying to retrieve epigenetic markers from GBS data, but it's possible. So, taking your reads and mapping it back to the reference genome is not exactly accurate, right? Because you're basically just saying, I'm taking this sequence, I'm matching it to the reference, and if it matches 100%, it's derived from that region, right? But I'm also looking for cases where it doesn't quite match, right? And I want cases where it doesn't quite match because that's how I discover my SNP, right? But you can also have a lot of problems because what about if you have duplicate sequences coming from 2 regions of the genome? You might end up taking 2 group of reads coming from 2 regions of the genome and mapping them to one single location, right? So what happens? You've just collapsed 2 different SNPs into one SNP. And that will lead to error, right? To lead to error because you're not going to get the accurate genotypic data. And 2, you're not going to place that SNP in the correct genomic location, or at least for the other copy, right? You also have cases where your samples might have a high sequence divergence from your reference, right? So typically, if you're working on con, right, you map your con reads to a con reference genome. But sometimes you don't always have a reference genome within the same species, right? So you might decide, okay, I'm going to use a reference genome of a different species, but within the same genus, right? Now you're going to start having more divergent sequences, right? And that becomes a little bit more problematic for alignment, right? Sometimes even within the same species, you could have a lot of structural genomic rearrangement. And this happens a lot, more than we'd want to admit, where you could actually have huge chunks of sequence missing from a sample within the same species, right? It's good to just know those things, right? So that when you derive your SNPs, you take that information with a grain of salt, right? You don't believe it 100%, and it sort of helps you in how you interpret your data downstream. If you're trying to do SNP calling in a polyploid, which typically people try to avoid for obvious reasons, so you might have a case where you have wheat where it has 3 subgenomes, right? And typically what people do is they look for regions in the genome that is subgenome specific, right? So you want to genotype your SNP in one of the 3 copies, right? It's present in this subgenome, but it's absent in these 2, right? So you score it like a diploid. And if you have a sequence that's present across all 3 subgenomes, what do? You ignore it, right? So imagine if 75% of your genome have those common sequences across the subgenomes. You're basically throwing away 75% of the genetic information in that genome, right? There are new tools to actually go in and recover what's a 2x. Let's say you're looking at a hexaploid genome, right? That's 6 copies. You can effectively go in, map your reads, and say, this read maps to a single subgenome. I'm going to call it 2x. This read maps to 2 subgenomes. I'm going to call it a 4x. And this sequence maps to 3 subgenomes. I'm going to call it a 6x. Does that make sense? At the end of the day, you can also retrieve the proportion of the reads that map to your reference. So in this case, you're seeing about 95% of your sequence reads mapping to the reference, right? But when you make your library, you do spike your library with what you call the 5x. It's a viral genome. And you put about 5% of that into your library. So effectively, you're demultiplexing and recovering pretty much all the reads you've sequenced coming from your sample, right? Or at least close to it. right? Because even though you're spiking with 5%, it might technically be like more like 3% or 6%, you know, depending on how good your pipetting skills are. And again, we're showing here where it's mapping to the 6X genomes and where it's mapping to unique subgenomes. On the right, you also have different performance with different alignment tools, right? So you could be using BWA or BWMM or Bowtie or Nova Align or Star. They all do perform a little bit differently. But typically, what most people use is the BWM because, as you can see, it does a better job, right? So if you look to the top left, you're looking at cases where you have SNPs, where you're trying to recover a SNP, right? You do see that the Star Aligner performs better, right? The BWMM is in yellow, right? But then when you go into the SNPs that are incorrectly assigned, you also see that the Star has a slightly, well, significantly higher false positives, right? So even though it's recovering more, it's also making more mistakes, right? And when you look at BWMM, you can see that the error rate is quite low, right? You could also look at cases where you're looking at insertions for things in the middle. You see with insertions, BWMM outperforms every other person there, right? And then when you look at the error rate, the error rate is also quite low. So when you're doing alignment, it's not a typical alignment you're doing, for example, where you're doing a blast, right? Where you just take a sequence and you blast it against another sequence. When you're doing alignments with SNP calling, the first thing you want to do is you want to index your genome. And you want to index your genome for obvious reasons. It is faster when you index your genome. It's more efficient, right? But one key factor is that when you align those reads to your genome, you're tracking the position of every single base in your read, right? So if you map everything, your 100 base pair sequence reads to the reference, you can basically say position 1 aligns to chromosome 1, position 1,056,000, right? So that at the end of the day, when you do your SNP calling, you can literally name your SNP with the chromosome number and the physical position in which it exists. Does that make sense? So I'm also showing you a slide here. So besides just doing your sequence alignment, so you do your sequence alignment, you get what you call a SAM file, you make a BAM file, you do some data wrangling before you actually do your SNP calling, right? So we're gonna skip through all that data wrangling in between and go to the actual SNP calling, right? So you could use different software or tools, right? You have what you call the Freebase. I'm sure Meg covered this, right? You could use Freebase, you could use GATK, Platypus, you can use SAM tools. There are a lot of tools out there. And here we're just comparing what is sort of the popular, well-used tools. And as you can see, the Freebase and over here, you can see that Freebase and GATK sort of outperforms the other tools. And that seems to be a consensus, just based on the way that tool retrieves the SNPs. We can go into that a little bit deeper. Okay. So we'll stop here. You're going to do some hands-on, and we have about maybe 1 or 2 steps in that pipeline that'll take about 20 minutes or so. So let's get that started, and then we'll come back to the lecture. All right. Questions? You understand everything? So questions?
It might be helpful. So I use the five samples, right? We could draw something about the 2x squared. So I don't know. We don't have much board space.
What do you mean?
Also,
Okay.
Some way to visualize.
All right. Wow. Ryan is setting up. So what you're going to be doing today will be using, so we have 5 samples. They are sweet potato samples. It's an hexaploid, and it's a segmental allopolyploid, which means it was derived from 2 species or 2 individuals that hybridized and combined. One was a 2x and one was a 4x, and they combined to form a 6x. All right. And we specifically using these data sets to drive home some points, right? Now, the possible ways to do the alignment to a reference genome would be to do a non-reference based approach, which we're not going to cover in this class. But I just want you to know you can also do SNP calling without the reference genome. Right now, it's a tough task, but it's possible. You could also use the ancestral progenitors of that polyploid. Remember I told you that sometimes you can use a reference genome derived from the same species, or you could go one step backwards and use something that's not the same species but within the same genus, right? So what we're going to do today is we're going to use a diploid reference genome that's in the same genus as sweet potato to do this SNP calling. So technically, you have the 4x and you have a 2x, and they're combining the same genome. That's a 6x. Right, so you're going to get a lot of 6x sequences. Why? Because these things have to be somewhat similar for them to hybridize, right? So you'd expect that most of your sequences will be 6x, but you're going to have unique sequences that are unique to the 4x and are unique to the 2x. Because it's only a 1-hour lab session, we're going to pretend everything is a 6x, right? And we're going to take that FASTQ file, which we've cleaned, the same way we did a lab session on Monday. We've cleaned it, we've trained it, everything is okay. What Ryan has also done is he doesn't want to give you a 20 gig file or FASTQ file that would require days to run. So what he did was just subsample the first 1 million of each of those 5 individuals, right? So you could think of it that you're subsampling just 1 million, which means you'll be reducing your read date because you're just sampling randomly, right? And we're only doing that because we want you to do this under 30 minutes or 45 minutes.
Yeah, it should still produce results though. All right. Does everybody remember which computer they logged into last week? Awesome. So, yeah, okay, awesome.
And you need to know, because if you don't, then you need to create a new folder.
Yeah, and if you don't remember, if you need to make a new directory, that's fine too.
Or better still, If you don't remember, choose one of 26 or 50 and see if you have a folder named after you. If you don't have a folder in your name, then you know you're on the wrong computer. Log out and log back into the other one to find where you are.
So for right now, we'll just, I'll have us just skip past this part about downloading the images. I still do want to do that, but for now, we want to go ahead and start getting our alignment going, just because I think the first part takes 9 minutes and then there maybe 20 or so. So.
And the password?
Oh, yes. Okay, so yeah, it's, guest at 622.
And I can, oh, you do have the paper, so you know what you're doing, right? I can also write it on the board. So the first thing you're doing is what, indexing?
Yeah.
Okay, and Ryan will tell you what's peculiar about the way we're going to be doing the indexing today.
So,
again, because of time constraint.
So go ahead and see if you can find your way into your project folder. Can you guys read this? Does this need to be a little larger, or do we need to turn the light down a little bit on this, or what's it like? Okay. Okay, so if you found your project folder, we're gonna do like we did last class, and we're gonna copy and use that R argument, and we're gonna copy the snip pipeline folder into our current folder. What's that?
You can make it bigger.
Oh, that's really hard to, yeah. So
is it better?
I can actually point to it today too. That's good. All right.
Better?
All right. So if you were able to successfully copy that, you should see that you have hopefully Mondays. If not, that's not a big deal. But you should have the snip pipeline directory. And let's go ahead and change into that directory. And so I don't know if do you guys know how to use like the ls command, the list command? Yeah. So sometimes I use the L option just to kind of list things a little bit more orderly and get more information about them. But I wrote down this directory structure on your sheets just so you kind of have a little bit better idea of what's going on. So that's what's going on right here. So we have these 4 folders within Snip Pipeline: the reads, ref genomes, scripts, and tools. And if you want to, you can actually change directory to the ref genomes. So this is where we have this is what we're going to be using as our reference genome for the alignment that we're about to do. So we've got Trifida, which is, oh gosh, this is not working now. What I did was I took this entire reference genome, and for time's sake, I also just took the 15th chromosome. But this is a characterized, well-known, diploid reference sequence that we can use. And
it's called Icomia Trifida. It's one of the diploid ancestors of SWIFT2.
So,
which you know.
So we have just that chromosome. This is going to be our reference genome. So that's where that's stored, and that'll make sense in just a second, hopefully. But if we go back a folder and change into our reads folder and list that, these are going to be the 5 Ipomwea batatas samples that we're looking at, and these are all hexaploid. So we're going to try to align them against this other reference genome, which is a slightly different genome in some ways. So we have these 5 samples, and we have this text file, which some of our scripts are going to talk with. And if we just look at that text file, it's up there. It's just a list of the names of the FASTQ files to look at. So can back out of there for a second. So we want to get into our scripts folder. So this is where everything is going to happen from this point on, is just the scripts folder. So it's just me having us go one step at a time, basically from the back side of your paper. We're going to go through this pipeline, and we're going to start out by creating an index. So that 15th chromosome that I just showed you, that sequence, to speed up the alignment of all those individual reads to that reference genome, we have to first index it. So that's going to make it a lot easier for that alignment to happen. And I always like to look at some of these scripts before I run them. So if we look at the first GBS underscore index, this is going slow. What it's doing is it's using the BWA tools, and it's creating an index of that 15th chromosome. And I don't think it's doing anything else too fancy at this point. So let's go ahead and type in bash, and then what do I have here? The GBS index shell script. And I think this should actually be pretty fast. Has anybody already run it? Never actually looked to see if you can just open up the index and look at it as a text file, or if that's dangerous to do. So if you were to go back into that reference genome folder, the directory we're just in, you can see that it's now no longer just the full reference and the 15th chromosome. We now have a bunch of index files that were produced by BWA, and these are going to be used by all the subsequent steps. Oops. Okay, so we have our index. I'll go ahead and open up the next script that we're going to do. So the next script that we're going to run is the actual alignment. So if we look at that, there's actually a lot that's going on in this shell script. So it's creating a projects directory to store all of this output. So that's what the make directory up there is doing. And then it is running a lot of tools. So this looks actually kind of messy. So I've written on the paper sort of some of the things that are happening. But what we're doing is we're taking all these lines that we have here, all these different commands that are using like GATK and PCARD tools and all these other alignment tools, and we're creating another shell script. So I showed you that key snip call file earlier, just the text file that had the names of those 5 hexaploid genomes that were, or reads that we're interested in looking at and aligning to our reference genome. So for each one of those file names, it's creating all of these commands 5 times.
That way you're not typing one sample at a time. You automate it.
So if we go ahead and run this, actually this one just takes a second, I believe. If we run it, it happened super fast, and we can open up the file that it created. It's called pre-snip calling. So this is going to be one of the ones that takes just a little bit longer to run. So you can see we went from something that looked kind of messy and confusing to something that's messy and confusing times 5. So it's all these different commands that are being run for each of our 5 samples. And it's going to align each one of them for the reference genome. Oh, yeah. So a lot of these, let me pull that up again, even though it is very hard to see. So with a lot of programs, you can specify how many CPUs to use on your computer. And so we're actually able to specify these commands either with some of these Java tools, we can tell them manually to use 2 CPUs threads. And then I think, oh yeah, and so sometimes you'll have like a -T or something like that. A lot of tools will let you multi-thread. Not all of them will. You just always have to look into the help file to see what kind of commands or what specific thing you have to type before. Yeah.
I'm just asking a simple question. How come you use script
Well, so that that script actually it contains a for loop within it, I guess, if that makes sense. So let me go back to that original GBS align. So within here, we're actually, oh, it's so hard to find. If you look up along here, if you go up to the 5th line down, we have an awk command, which is, this is all very wordy. There's a lot of typing that went into making this part of this script. But at some point, oh gosh, it's so hard to find. Yeah, it's cut in half, making this really useful. At some point, that awk command is looking into this keysnip call file, and it's pulling sort of like a variable, like a wildcard variable. So we're using it for 5 different samples. So even though it took a little while to type up all of this, if we had 500 samples or more, you can run this exact same script on, as long as you have that little keysnip call file, you can run this on as many samples as you want, and it just creates this incredibly long shell script that you can run pretty effortlessly. And you can use it for a lot of different projects.
One other reason why we're not using a for loop, so even though this is not technically a loop, but it automates the process, right? If you're using a loop, then you need to specify that you're going from 1 to 100, right? Or you can list what you want to do, right? The beauty of this is you can define the name of your sample upstream and track the sample, right? If you don't want to use the off, you just say, okay, I know my sample goes from 1 to 500. Loop through 1 to 500. But at the end of your analysis, you want to go back, and we did this in the first class, right? Where we demultiplex. And we actually did a for loop going from 1 to 24 and 1 to the 16 for the reverse, right? And then at the end of that process, we can go assign 1 to the sample, right? So imagine if you had a file that had like a serial number 1 to 24, and that serial number matches the sample, right? You can technically do a for loop and then match that serial number to the sample. But if you do this, you don't need to do that. You can go anywhere during that pipeline and identify the sample. If you are interested in a specific sample, you can go anywhere in the field of that pipeline and say, I'm going to go out this sample. And you don't have to go reference. Right? Do that means.
So if you haven't already, we'll go ahead and run the pre-snip calling shell script. So this is going to take anywhere from 10 minutes to more than 10 minutes. So we'll just see how that goes. Has anybody run into any issues with this pipeline yet? Or? Awesome. I was impressed that you guys all, I went up to the lab computers after Monday and I was like, hey, everybody has like a folder and it looks like people were getting results. So good job. All right. So at this point we're running the alignment. Is this an okay place to pick up back with the collection? This is actually performing all of the alignment. So this is going to take a little while.
All right, we'll come back to it. if at some point during the discussion, the alignment, your alignments are done, let me know. I'll stop and then we'll go on to the next thing. Any questions? Are we all on the same page? I can wait if you're still typing your command in. All right, cool. Okay, so you're doing your alignments right now. The next thing you're going to do is going to be what we've tagged a pre-snip calling, right? So you're going to generate a SAM file, you're going to convert to a BAM file, and then you're going to index that BAM, and you're going to do a few multiple things that Ryan is going to go over, but as you're going through each of those processes, you're sometimes indexing your BAM, and sometimes you're sorting your BAM file. Because it needs to organize all that file. Each time you run a process, it shuffles these things, it shuffles the file a bit, and then you need to resort it and reindex it so that you're pointing everything in the right direction. All right. Once you're done with the pre-snip calling, you're going to perform your actual snip calling. And I think at this point, most programs will generate what you call a VCF file. And the VCF file does look scary. It's got like all this information, right? Some of them are empty information, like, do you have phasing information? And phasing basically means if you have two SNPs, what configuration are they on the chromosomes, right? Is it AC, AC, or AC? Okay, let's do something else. AC, GT, right? Is it AC, GT, or AC, TG? Does that make sense? So that's what you call the phasing. So you could have information like that if you've processed that. And the first thing you'll actually see in your VCF file is a description of all those parameters, right? So if you look up there, you'll see, okay. So you see GT, and it gives you a description of what GT is, genotype. It gives you the genotype quality. It tells you what the read depth is, how many reads mapped to that region, right? It also gives you the allele depth. And okay, I don't think I see the description of the allele depth here, right? No, it's not showing it here. But you'd have that information as well, right? So you're seeing a SNP, C, and A. It can also tell you how many reads are mapping to C and how many SNPs are mapping to A, right? If you come down, you get your chromosome number, the position. It will give you the ID if you actually want like a unique identifier for that SNP, right? So you could name it as, say, chromosome 1, and then the position or something like that. It will tell you what the reference allele is, right? So what that position is on the reference genome, and it will tell you what the alternate is. And the alternate is the polymorphism that's not on the reference genome, right? And that polymorphism could be an A, right? So in this case, it will be, let's use something simpler. If it's C, then the polymorphism is CT. But you also have an allele called CT, right? So basically, you have an insertion of a base there, right? The simplest you can have is just two possible alleles, right? A or G, right? And then you could have a situation where you actually have a SNP, but that T is completely missing in your sample. So you call it a deletion, right? It can tell you what the quality score is, if you passed filter, you know, the format it's showing here is your GTDP. Usually the files you generate are a little bit more complex than this, right? It gives you, you know, likelihood scores and phasing, you know, but this sort of, you know, just some of the basic information you want in a VCF file. Okay. So again, you have your SAM file format. We won't go over this because I feel like you probably talked about this in your previous classes, right? And then you have your BAM file. I'm just gonna check and see if it's still running. All right. So the first thing you want to look at once you get your VCF file, right? You want to extract some key information. What type of information do you think you would need? Well, it depends on what you want, right? But you definitely want to see what the genotype is, right? That would be the first thing. Sometimes you might need to retrieve the read depth because you might want to use that for filtering. Remember, we talked about how you need multiple reads to capture an heterozygote genotype, right? So you can say, if I only have a read depth of 1, I'm going to throw away that SNP because that's not sufficient to capture an heterozygote genotype, right? You could say, well, I want to see 5 reads to confidently call this SNP, right? So you need that information. You have what you call VCF tools that can intuitively go into your VCF file and filter things for you, right? So you can filter based on read depth. You can filter and say, just pull out chromosome 1, right? You can do all sorts of filtering. You can do some other type of analysis using the VCF tools, using your VCF file. But what we're going to be doing following our SNP calling for this lab session is we're actually going to use VCF tools to pull out the genotype information, the read depth, and then we're going to pull out information on the reference allele and the alternate allele. Because sometimes once you get your SNP, or if you're using your SNP for a particular genetic analysis, you might want to know what that variant is, right? You might want to know if it's an A or C, or if you want to design a primer from that. Okay, you might want to go into the sequence context. So once you've pulled out all that information, the first thing you can do is just to get a feel for the distribution of the read depth in your data set, right? So if you look here, you're basically going from 0 to, you cap it out at 1,000, right? And you see a fairly uniform coverage across the samples, right? And it's sort of tight, right, within that region. But you're going to also have cases where you're going to have what you call outliers, those red circles, right? So, you know, remember we talked about optical duplicates or PCR duplicates? You could start capturing, you can actually see cases where your PCR duplicates, if not removed, you know, will come out in these outliers, right? Because you're tending to have more of them than what you'll get for the other fragments on average. Huh?
Can you compare image that looking at it?
Well, you don't know. You're just in the distribution of your read depth. You're hoping that if you're sequencing everything at about the same rate, you should get, if you get 20x coverage, you should get a 20x. Everything should cluster together at 20x, right? But you're going to have variation in terms of coverage. Why? Imagine if you have a duplicate gene, a paralog. So let's say you sequenced everything at 20 cycles, right? You did a PCR, generated 20 copies. Well, if you have a duplicate copy of that sequence, it's going to be a 40x. if you have a 100 duplication of that sequence, 20 times 100 will give you 200. Is it possible to get a 100 duplication? Is it possible to duplicate a sequence 100 times in a genome? Did you say yes? How is that possible?
It's especially for counts, a lot of
Repetitive elements. Actually, animal genomes have it too. Transposons, retro elements, right? They are so similar. Some of them are duplicated by being copied and pasted, right? So you copy them, you paste them in somewhere else in the genome, copy them, paste them. Sometimes you call them what? Jumping genes, right? Remember this? Right, so you could have a case where a sequence just has so many duplications across the genome, right? So even though it's not technically an optical duplicate, you're going to get so much enrichment of that sequence, right? And that's what's leading to all this, right? So you want to be able to go into your data and remove those things, right? So it's not enough to just generate your VCF file. You have to be able to say, what are the useful SNPs that map to a unique locus in the genome, right? And that's an entirely different problem in the pipeline. Something else I've showed you, right? If you're looking in an exploit, remember we said you're going to have like 2x copy, 4x copy, and the 6x, right? Well, so what we've done here is to map all those SNPs after filtering, actually, right? And as you can see, the green are things you've genotyped as 6x. They pop out at about 500, right? And then things you genotyped at 2x pop out at about 100, 125, right? Which sort of scales, if you'd expect a 1 to 6, right? And then the pink stuff is somewhere here, it's sort of slightly shifted, which you'd expect to be as 2 times the copy of a 2x, right?
Why the 4x?
Huh?
4x.
Well, if you zoomed in here, you would see it better. Why you not having a lot of, why is it that the 6x are so much more copies than the 2x and 4x? Because they are very closely related species that generated that polyploid, right? That's why they're able to hybridize. They're very similar, right? So they do have the same copies of genes between them, but every now and then you get the regions in the genome where you have sequences that are unique to them, right? And we know this is about 3.5% for the 2x and 3.7% for the 4x.
So that means if you sequence your sample, maybe you cannot sequence too deep. That's less than 50.
Yeah. So it depends on how you make your library. If you digest your genome with a frequent cutter, you're going to get lots of fragments, right? But if you digest your genome with a rare cutter, you're going to get small fragments and then a lot of big fragments, right? And then you size select, you're going to enrich for the small fragments. So you're basically eliminating some parts of the genome. And that's why you call GBAS, or sequence-based genotyping, a reduced representation sequencing. So you're just capturing about 0.05% of the genome or less, if that makes sense. So you're only sequencing random small segments of the genome. And then, depending on how much of that if you're sequencing a 0.05 portion of your genome, you're going to have a lot of very good coverage, right? But if you sequence the whole genome, what happens to your coverage? You reduce your coverage, right? So if you spread 250 million reads across 3 gigabytes of data, you might end up with a 1x coverage. But if you spread your 250 million reads across 0.0001 gigabytes of sequence, then you increase your coverage, right? And that's why GBS is cheap. Remember, we talked about this on Monday. So some of the other things some of the other images you want to generate, right, is the genotypes, right? So you could have a chart showing you the multi-allelic and biallelic genotypes. Can someone tell me what a biallelic genotype is? Only has 2 states, right? So it's either 0, 1, 0, 1, right? But you could also have cases where you have a 3rd allele, a 4th allele, a 5th allele, right? So if you're in a diploid, what's the maximum number of alleles you can derive from an individual? 2. Right? If you have 2 individuals, what's the maximum number of alleles you can derive? 4, right? But as you add more individuals, most of those alleles are going to be shared in that germplasm, right? So your ability to detect a 2nd or 3rd or 4th, as you increase the number of alleles, the frequency of those alleles drop. They're basically more like minor alleles, and that's what you're seeing here, right? So in most organisms, what you should be seeing most of the time are biallelic markers, 0, 1, right? 0, 1, and then all the other possible states will be minor. And what you do normally for most genetic analysis, it's easier to work with biallelic markers. So typically people just filter all this out. Not because they are minor, but because sometimes it's difficult to work with multi-allelic state markers, right? So people tend to remove it. And then you end up with something like that on the right, where you're just seeing the 0 allelic state, which is the reference, and the 1, which is the alternate. And most of your SNPs should fall into that.
Did everybody's alignment finish yet?
All right, we'll take a break.
Yeah, now it's the longest step, so I'm sure we get that. All right. Did everybody say there were no warning messages? Okay, cool.
I did get one that said unable access jar file.
How many people have that problem? If you're done, can we see your hands? Well, without if you're done without any error, so how many people got error messages? 2. So just rerun it.
Well, it might be, I'm going to guess that maybe that has something to do with the way that the folders were copied. I'm not sure. Well,
I mean, she should be copied in the same way.
Yeah.
So sometimes, because you're running multiple Java jobs, sometimes they compete, right? It's like, so I think that would solve it. Just rerun it.
Sorry about that. All right. So the next step that we're going to do is, so we have our alignments at this point. So we have like the positional information for where those reads are mapping to our reference, our previously indexed reference. So now we're going to run GATK. Actually, let me look at that file before we run it. Just look really quickly.
Well, you're doing it. You just finished the alignment, right?
Yeah.
Or the pre-SNP calling.
Yeah.
Okay, can we look at the pre-SNP calling before you do the GBS call?
Oh.
So if you look at that, right, so you start off with your alignment and then you generate the SAM, right, and then you make your BAM and then you sort that BAM, right? You index it. You should have covered this from your previous class, right? What you do during this pre-SNP calling process, because I'm thinking about skipping it, but I just want to highlight something, which will be the summary after the first BAM. And you go into the summary folder to see the proportion that match to the genome.
Yeah. If you guys remember, we created that. One of the scripts created that batatas folder, just one back. Oops. So if we look in there, there's a subdirectory called summary. Let me open one of those.
Okay. Right. So if you open each of these files, basically gives you a summary breakdown of your alignment. Right. So if you just open any one, right? So the first one tells you how many of them passed QC. Right. So it's telling you everything passed QC. That's good. Right. The next one tells you, do you have duplicates? No. Right. And then the next one tells you how many you were able to map to the genome. How come we didn't map everything to the genome? Or we didn't map the 95% I alluded to earlier? If you're only mapping 11% to the genome, that sounds like that doesn't sound like a lot. Does someone have an explanation? Why are you only mapping 11%? Right? You're only doing chromosome 15, right? For obvious reasons.
Yeah, and the reads that we're trying to map to chromosome 15, they were just kind of think of them as being randomly drawn from the actual genome. So,
but considering chromosome 15 should be one of the smaller chromosomes, what would you expect? What proportion would you expect to map to chromosome 15? Math. You have 15 chromosomes, and usually so we have 15 chromosomes. Okay, sorry, I didn't tell you how many chromosomes we have. So you have 15 chromosomes in sweet potato, right? And you have a 16th pseudomolecule. Things you can map to the physical chromosome, you just throw them into this giant pseudomolecule for things you can map, right? And just stitch them together without any particular order, right? So we call that chromosome 00, right? So technically we have 16 pseudomolecules. Let's assume all those chromosomes have the same size. How many of those reads would you expect to map to chromosome? Percentage-wise. So why are you getting as much as 11%? That's like twice as much as you'd expect to get. I sort of talked about it in the last two slides. Number. So think about it, right? You have 15 chromosomes. You're going to have duplicate sequences. You're going to have repetitive sequences that are duplicated, shared across those chromosomes. Right? Do I need to explain this anymore? Or do you get it? So I'm only bringing that up because I'm trying to drive home a point. Paralogs are a big deal when you're doing snip calling. It's a huge problem when you're doing snip calling. And typically, people just avoid it. They find a way to remove it from the final set of snips. Okay?
Yeah, we should go ahead and do this final. That's not the final step, but this is the longest step. So the GBS call script. Let's go ahead and do that. There we go. So hopefully this won't have any competition between computers on the same the same computer.
Using GATK. Could use FreeBase if you wanted to, or samples, or variant scan.
As long as one person's computer gets it. So I could open up another instance and you could capture the images at some point too. But yeah, but we can do that at the end of class too.
All right, I'll just finish up. All right. So talking about, and it seems I'm like beating a dead horse, and I'm emphasizing this whole concept of paralogs, right? What I'm showing here, okay, let's look at this. Talking about coverage, right? So this is something Illumina put out there, right? So basically they're like, if you're doing snip calling, this is the probability of your snip call accuracy, right? So they're basically saying, if you have one snip, the probability of catching, calling the right is like 0, right? And we know why. If you have 2, 3, so they're basically saying you need to get up to like about 5 or somewhere between 5 and 10 to really start recovering your genotypes accurately. I don't think it's that bad, right? Why? Because even if you have just one read, you should be able to capture an homozygote, right? So it shouldn't be 0. Does that make sense? If you have only one read, and that genotype is an homozygote, it should be sufficient to call that SNP, right? So if you're in an organism that's an inbred or a double haploid, do you know what a double haploid is? No. Do you know what an inbred is? So basically, if you take an individual and you keep selfing it, at some point at the 7th or 10th generation, about over 95% of that genome will be homozygote at every location, right? So if that's the type of genome you have, then you only just need one read to see most of your SNPs, right? But if you're looking at a highly heterozygote genome, right, that has that variation between the two states, then read depth becomes crucial, right? And as you approach a higher coverage, you sort of flatten out to like almost 100%. So what we've done here is a situation where we're gonna try and remove all the paralogs, right? So there are 2 ways you can remove the paralog. You can physically go into the genome and identify all the paralogs in the genome. But that's sort of tedious, right? So one thing we've done, which I won't go into much detail, is we did something called resampling, where we took the parental individuals that created a population and we sequenced them really deep to a few thousand, right? And then we started subsampling, right? And from the subsampling, right, we capture one read and we say, what's the accuracy So let's look at the top. So when we subsample, on average, we capture one read. And we say, what's the accuracy of capturing that genotype accurately? 2 SNPs, 2 reads, 3 reads, all the way to like 500 reads, right? And as you can see at the top there, you do see quite a bit of noise. But as you approach, so that's a diploid. As you approach somewhere here, which is about 15 or 20, which is what you have here, it's sort of comparable, right? If you compare between this and that, right? So without filtering your data, you do need somewhere close to 20x coverage to capture an heterozygote accurately in a diploid. So what we did was we went back in and we said, whatever has a very bad accuracy, we're going to remove it. We're going to filter it out. And once you filter it out and you redo the same thing, boom, you realize that you only need just about 6x coverage to capture an heterozygote. And at that point, it's clean. The point I'm trying to make is these 2 data plots here include paralogs, right? And that just creates noise in your data, right? So think about it. If you have a duplicate, it starts behaving like a 4x genotype. If you have 3 copies, it starts behaving like a 6x genotype, right? Because you're collapsing 2 sequences here, or 2 states, and you're pulling everything together, right? It increases the complexity and the ability to consistently capture all the allelic states across all those genotypes. And that's why it's sort of messy. But if you clean it well, everything comes out clean. And that's the same case you have in here. If you're doing it in a tetraploid, you see at the top there, it's really messy. At the bottom here, things get cleaner. And when you're looking at hexaploid, it gets cleaner. But you also realize that the amount of read depth you need to even capture a single locus in a tetraploid is higher than a diploid, right? So you're looking at a 6x for a diploid, about 35x for a tetraploid. And in an hexaploid, you need about 100x to approach 95% confidence. Questions? So if you notice throughout this lecture, one thing I've been trying to drive home is to understand the limitation of the technology, understand the inherent error in your data. All right? And possible ways to fix it or to extract what you need. Questions? Discussions? Something we didn't talk about or that we missed? Yeah, 20%. So we're about 20% of the way done. And the cool thing with GATK is it helps you track that, right? So if you look at, there's a column there that gives you the percentage. Okay, I'll stop talking and I'll let you process for a bit. And then maybe before this is done, I will talk a little bit about GATK. And so it's basically going to be GATK freebase and why those SNP calling tools are the best and why you should be using them. You can look through the slides if you have questions. And by the way, were you able to copy the output files from the last class? Okay. So do you have like a file sharing client? The secure copy. Okay. Did anybody try using the secure copy with your PC? Well, she has a Mac. Did anybody try doing that with a PC? Yeah. And it worked? Where we are, 30%. Okay, I'll quickly talk about the GATK. And what I'm going to be saying about the GATK applies to some other tools like FreeDays. And the reason why they are superior to some other snip calling pipeline. So, one, they give you the option to do any level of ploidy, right? Some tools, you can only do a deploy, right? They offer as much as a tetraploid, a hexaploid, an octaploid if you're working with something like Strawberry. Another cool thing about GATK is, so instead of just taking your reads and aligning it to your reference and calling the snip, it does a little bit more than that. It takes those reads, align it to the reference, takes all those reads that align to the same location, and does a local reassembly, right? So it basically takes those reads and does an assembly, and then infers how many haplotype you have. Do you know what an haplotype is? And that's why it's called an haplotype caller, if you look up there. So an haplotype is basically, so let's say you have a stretch of sequence and your SNP on one of that sequence is ACT. But you have another fragment coming from that same region that's GCC, right? So you have ACT, GCC. You have two haplotypes, right? So even though they are coming from the same region, the SNPs, right? You have a sequence of SNPs that identifies them as being different from these guys, right? So you could have an haplotype that is ACT, you can have an haplotype that's GCC, you can have an haplotype that is ATT, right? Now, so each of those base calls is an allele, right? And you're sort of combining them, right, into an haplotype, right? So even though they're all coming from the same region in the genome, they have these different arrangements of these SNPs. And the reason, one of the reasons why you identify them with that sequence of SNPs is because they tend to be inherited together, right? So if you think of the genome as being reshuffled every time, right? An haplotype, they stick together, right? So that ACT, whenever you reshuffle the genome, that ACT goes together. That GCC goes together. That ATT goes together, right? So they are almost behaving like alleles, basically. They are like behaving like one unit of, sequence that's always moving together, right? And you can track them in a population. You can say, this population have this string of SNPs that are always moving together. All right? Now, that's important when you're doing a SNP calling, because you can basically say, if I'm doing a single sample SNP calling, so I'm doing SNP calling on just one individual, and that individual is a diploid, how many haplotypes do you expect? 2, right? One chromosome, another chromosome. This chromosome has specific arrangement of the alleles. This chromosome, right, has specific arrangement of the alleles, right? Does that make sense? So they are like units within the genome that are distinct, right? So those are 2 haplotypes. So if you're calling a diploid in just one, calling a SNP in just one individual, you can basically tell the haplotype caller in GATK and say, if you see anything that's more than 2 haplotypes, get rid of it, because it's probably a paralog. We're coming back to the old story of the paralog again, right? But it's a little bit more complex once you have a larger population, right? Because now you have a chance of having more than 2 haplotypes, right? But you could still peg the number of haplotypes, right? Because it doesn't matter how big your sample is, your chances of seeing 2 haplotypes is high, but your chances of seeing 4 haplotypes is lower. Your chances of seeing 6 haplotypes is lower, right? So the only reason why you'll be seeing 10 haplotypes, for example, is probably because they are paralogs coming from multiple regions in the genome, right? So the GATK type of SNP callers are also robust in that way. And that's just another layer of filtering that helps you to generate, a higher proportion of quality rates. Yeah. So these 5 samples, you'd expect more than 2 haplotypes. So where you peg it will be arbitrary, right? You could say, I'm okay with 6 haplotypes. Don't look more than, or I'm okay with 4 haplotypes. Give me anything more than 4 haplotypes. But again, you can understand that it's really a continuum, right? There's no one threshold that would work for every single locus in the genome, right? %60. So one thing we talked about was probably to have started your pipeline at the very beginning of the class, but that was on the retrospect now. Well, we're more than halfway done. So once we're done with this, you're going to run an R script. And I'm not going to bug you with the details in the R script, but what we're doing with the R script is using quantitative genetic parameters to even extract higher proportions of quality SNPs. So if you're in a biparental population, do you know what Mendelian segregation is? Huh? You know what Mendelian segregation is, right? So if you have two parents making progenies, if it's homozygote, heterozygote, what proportions of genotypes do you expect? 1:1, right? So if you have big A, small a, and small a, the only possible genotypes you should be seeing is going to be big A, small a, big A, small a, sorry, small a, right? And they should be in a segregation ratio of 1:1. So what I can do is I can go into my multi-sample VCF file or genotype file and say, look across all the individuals in this population for this one SNP. If the ratio of small a and big A deviates from 1:1, that's a bad SNP. Get rid of it. It really works like magic for filtering.
But don't you worry you're throwing out some good SNPs though?
Well, it depends on what you're going to use it for. So if you're using it for, say, making a genetic linkage map, well, you need to get rid of it in the first place, right? But again, it's also a question about false positives and false negatives, right? If you had to jump into a rocket and fly to the moon, and someone told you, well, you know, you have a 50-50% chance of surviving, would you jump in? No, you wouldn't. So my point is, it's a better strategy to eliminate stuff, even if there's a chance that it's a false negative, right? And usually you get more than enough SNPs anyway. So why are you trying to hoard so much? You know, so it's better to get rid of the bad stuff so that you have a smaller size that is sufficient to get more accurate results, right? And the number of SNPs you want, you know, you can always shift your threshold, right, in a way to get the optimal amount of SNPs you want, right, without sacrificing accuracy. Another easy way to filter is if the genotypic expectation is small a, big A, and then all of a sudden you see big A. Ooh, what do you do to that genotype? I'm going to just turn it into missing because that shouldn't be happening, right? Right? If I see a big A, there's no way that's possible. So I'm going to make it missing or get rid of that SNP. I can make it missing if it's just happening maybe once or twice in 200 genotypes. I can say, okay, you know, that's a fluke. I can just turn it to missing. But if it's 30% of the genotypes having big A, then I know there's a huge systemic problem. So I can just get rid of that SNP. All right? So those are possible ways. And you know, those are some of the things we've coded into the R script to systematically go into your genotype data and look for those biological features, right, of your data and use that as a basis for filtering. So it's also going to use redepth to filter, right? So if you say, I want my SNPs to be at least 6, you can, what the R script will do is it will map a genotype to the corresponding redepth. If the redepth is less than 6, it will turn it to missing, right? So instead of just getting rid of everything, right? So what people, again, do typically is they just say, take the average redepth. If the average redepth is greater than 6, keep it. Well, the problem is an average is only an average, right? What about if 30% of your genotypes are below 6? Well, you've just, or 30% of your genotypes are 1, but your average is 6. Well, 30% of your genotypes are going to be wrong because their redepth is 1, right? So what we've done, used with the R script, is basically create a loop, right, that goes in and says, look at this genotype, go look at the corresponding read depth. If it's less than 6, turn it to missing, right? So do it iteratively. And then at the end of the day, after doing all that, you can go back and say, I only want a missingness of no more than 20%. If I have a snake that's more than 20%, get rid of it and keep the rest, right? So what you're just basically going to do is run the R script. Everything is automated. It runs, spits out your data, which you can view at the end of the day. If you want to look at the R script, be my guest, but it's a bit complex. But if you're interested in it and you want to know the nuts and bolts and how it works, you can reach out. And we're close. And the R script should run fast. I mean, it will run in like, I don't know, maybe 2 minutes.
What percent is everybody else at right now?
What's oh, yours is done? Oh, okay. All right. How many, how did you turn out to be faster than? So how many of you done? Oh, so you're like, okay. Oh, okay, you're done too. All right. So while everybody, the rest of you are still waiting on your stuff. Yeah. Right. So again, at the end of the day, you can get all these, all these message at the end of the day. Again, that basically, GATK does a lot of things. It goes in and tells you, basically gives you a feel for the quality of your data and its usage, right? And if you start seeing high values in that, then you should go back and think how you process the data.
All right. So I realize like class is basically over at this point, but let's go ahead and just run the bash underscore format, and then.
What does that do?
So I think that's just using the VCF tools to.
So
let me go.
Just extracting your GT, your DP. And they all alternate and reference values.
Yeah, and it's creating this header ID.
And that's using the VCF tools.
Yeah, and then have you all used Nano before? So I keep using less. That's what I use to open up my text files usually, just to really quickly look at them and not mess them up. But Nano is a great way to mess your files up, which we want to do with this snip filtering R script. So if you use Nano to open that, you can go down. We're going down to the, I think it's the 11th or so line, the set working directory. We're just going to change all those pound symbols in there to whatever your username is. Oops, don't do what I just did. Is everybody able to do that? So just change it to your name, and then is it Command or is it Control O on Mac? Let's find out.
Use the.
Oops, nope. It must be Control. Okay, I was assuming it was Command. All right. All right, and then just so after you use Control O and then hit Enter to save it, just exit out of there, and hopefully, if we run the R script command, it will work.
So you're using the.
So this is running the
command R script, and the beauty of that is it's going to be running your R script, and you're going to be seeing everything in real time as it's happening.
oh. It's actually giving me a warning message now.
Okay, remove 6. No, that's not a problem.
Okay.
So that's just for the. Okay, so it actually ran.
It created TIFF files.
Yeah, it ran everything. So that's faster than I thought actually.
So that just produced a bunch of TIFF files, which it's placed in the, I don't know if you guys can actually even read this, but within that Batatas project directory, within the summary subdirectory, we've got all of these pink TIFF files. So if you wanted to extract those on your own personal computers, you could use just the exact same commands that we have here. You'll just need to give the path to this summary folder, and then I would just use that little *.tiff just to copy the images only, so you don't get all this other stuff.
So some of the files here are stuff on the slide, right? So these two are the box plot, and these just like ordered based on the median, and the biallelic basically gives you the proportion of the genotypes. And one last thing you might take a look after is the minor allele frequency, right? So if you remember, we talked about how base calling errors can lead to a lot of minor allele frequency, and if you remove that up front, then you get an accurate distribution of the allele frequency in the actual population.
So I have the output and just to.
Right, you only go through the output, fast, and then, we're pretty much done with it.
Yeah, I think that's.
All right. So you can look at it closer. If you have questions, you can shoot me an email or talk to Meg. And we can leave the EPP622 open for the rest of the week. All right. Have a nice one.